Today we'll see the another experiment, that is the qualitative test for lipid. The experiment number 15, to study the qualitative test for lipid in a plant material. We'll see the principle as well as the procedure, requirements and result. First of all, we'll see the principle. The term lipid is used by the Blower in 1943. The group of naturally occurring substances which are insoluble in water but soluble in organic solvents like ether, chloroform, alcohol, and acetone are called as lipids. Chemically, the lipids are identified as esters of fatty acids. Alcohol and the lipids are also rich in hydrogen, oxygen, carbon as in the form of reserve food material, that is the fats and oil. Requirements: plant material, that is the soaked seeds of groundnut, glasswares like test tube holder, mortar and pestle, water bath, muslin cloth. Chemicals like sodium bicarbonate, 50% alcohol, then I2K solution, 10% NaOH, NaCl and copper sulfate. Procedure: first, take a 5 gram of soaked seeds or oil, grind in thoroughly with a 20 ml of distilled water. Filter the grinding mixture through a double layered muslin cloth. Use the filtrate to perform the test as shown in the observation table. First of all, this is the first test, that is the emulsification test. Take a 2 ml of oil, distributed sample or plant material, plus 4 ml of H2O, plus few drops of 6% sodium bicarbonate and shake well. In observation, the lipid is through a solution to form a emulsion. That means the emulsion, two liquids that would not normally mixed. In inference, you observe that is the formation of emulsion indicates that the presence of lipids. In a second test, that is the solubility test, take a few drops of oil sample in a two separate test soluble in a tubes. In a first test tube, add a 2 ml of water, and in a second test tube, add a 2 ml of alcohol or chloroform. Shake well and observe. The oil sample is insoluble in water but soluble in alcohol as well as the chloroform. In inference, we'll see the oil is soluble in organic solvents like chloroform and alcohol. While in a third test, that is the iodine test, you can take the few drops of oil sample plus add a 1 ml of iodine solution. In observations, you observe that the yellow color disappears due to the absorption by lipid. It indicates that the lipids, these are present in a plant material. After completion of that test, in the results, you note that in a given plant material, lipid content is present. Thank you.